Every second of your life, you are under attack. Billions of bacteria, viruses, and fungi are trying to make you their home. So our bodies have developed a super complex little army with guards, soldiers, intelligence, weapons factories, and communicators to protect you from, well, dying. For this video, let's assume the immune system has 12 different jobs. For example, kill enemies, communicate, etc. And it has 21 different cells and 2 protein forces. These cells have up to 4 different jobs. Let's assign them. Here are the interactions. Now, let's make this understandable. First of all, let's add colors to the jobs. Now, let's illustrate the cells. The central color represents the main job of the cell, while the surrounding ones represent secondary duties. Now the immune system looks like this. Now the interactions. Isn't this complexity just awesome? For this video, we will only talk about these cells and ignore the rest. So what happens in the case of an infection? It's a beautiful day when suddenly a wild rusty nail appears and you cut yourself. The first barrier of the immune system is breached, your skin. Nearby bacteria seize on the opportunity and enter your wound. They start using up the body's resources and double their numbers about every 20 minutes. At first, they fly under the radar, but when a certain bacteria population is reached, they change their behavior and start to damage the body by changing the environment around them. The immune system has to stop them as fast as possible. First of all, your guard cells, known as macrophages, intervene. They are huge cells that guard every border region of the body. Most of the time, they alone can suffocate an attack because they can devour up to 100 intruders each. They swallow the intruder whole and trap it inside a membrane. Then the enemy gets broken down by enzymes and is killed. On top of that, they cause inflammation by ordering the blood vessels to release water into the battlefield, so fighting becomes easier. You notice this as a very mild swelling. When the macrophages fight for too long, they call in heavy backup by releasing messenger proteins that communicate location and urgency. Neutrophils leave their patrol routes in the blood and move to the battlefield. The neutrophils fight so furiously that they kill healthy cells in the process. On top of that, they generate barriers that trap and kill the bacteria. They are indeed so deadly that they evolved to commit suicide after 5 days to prevent them from causing too much damage. If this is not enough to stop the invasion, the brain of the immune system kicks in. The dendritic cell gets active. It reacts to the signals of the soldiers and starts collecting samples from the enemies. They rip them into pieces and present the parts on their outer layer. Now, the dendritic cell makes a crucial decision. Should they call for antivirus forces that eradicate infected body cells or an army of bacteria killers? In this case, antibacteria forces are necessary. It then travels to the closest lymph node in about a day. Here, billions of helper and killer T cells are waiting to be activated. When T cells are born, they go through a difficult and complicated training process, and only a quarter survives. The surviving cells are equipped with a specific setup. And the dendritic cell is on its way looking for a helper T cell with a setup that's just right. It's looking for a helper T cell that can bind to the parts of the intruders which the dendritic cell has presented on its membrane. When it finally finds one, a chain reaction takes place. The helper T cell is activated. It quickly duplicates thousands of times. Some become memory T cells that stay in the lymph node and will make you practically immune against this enemy. Some travel to the field of battle to help out. And the third group goes on to travel to the center of the lymph node to activate a very powerful weapons factory. Like the T cells, they're born with a specific setup. And when a B cell and a T cell with the same setup meet, hell breaks loose. The B cell duplicates rapidly and starts producing millions of little weapons. They work so hard that they would literally die from exhaustion very fast. Here, helper T cells play another important role. They stimulate the hard-working factories and tell them, "Don't die yet, we still need you. Keep going." This also ensures that the factories die if the infection is over, so the body doesn't waste energy or hurt itself. But what is produced by the B cells? You've heard of them, of course, antibodies. Little proteins that are engineered to bind to the surface of the specific intruder. There are even different kinds of antibodies that have slightly different jobs. The helper T cells tell the plasma cells which type is needed the most in this particular invasion. Millions of them flood the blood and saturate the body. Meanwhile, at the site of infection, the situation is getting dire. the intruders have multiplied in number and start hurting the body. Guard and attack cells fight hard, but also die in the process. Helper T cells support them by ordering them to be more aggressive and to stay alive longer. But without help, they can't overwhelm the bacteria. But now the second line of defense arrives. Billions of antibodies flood the battlefield and disable lots of the intruders, rendering them helpless or killing them in the process. They also stun the bacteria and make them an easy target. Their back is built to connect to killer cells, so they can connect and kill the enemy more easily. Macrophages are especially good at gnoming up the bacteria which antibodies have attached to. Now, the balance shifts. In a team effort, the infection is wiped out. At this point, millions of body cells have already died. No big deal, the losses are quickly replenished. Most immune cells are now useless, and without the constant signals, they commit suicide, so as not to waste any resources. But some stay behind, the memory cells. If this enemy is encountered ever again in the future, they will be ready for it and probably kill it before you even notice. This was a very simplified explanation of parts of the immune system at work. Can you imagine how complex this system is? Even at this level, when we ignore so many players and all the chemistry. Life is awfully complicated, but if we take the time to understand it, we always encounter endless wonders and great beauty.
Alright guys, that was a Kyrgyzstan video. Awesome animations. They make tons of really cool videos. You should watch them. Now, they talked about the innate immune system there a little bit. They talked about like a macrophage, they talked about some dendritic cells, and they talked about a dendritic cell grabbing a piece of the pathogen that's invading and presenting it on a receptor on its surface. And then that then goes to the nearest lymph node and basically tells T helper cells and B cells, hey, we need to make some antibodies. And so that's what we're going to talk about today. That's really the quintessential understanding of what you need to get from this class, which is how do I take cells from the innate immune system and get them to present antigen or perform antigen presentation, as you can see here, to B cells or T cells of the adaptive immune system. That's really the job of the innate immune system. Yes, it defends you and like it's that first line of defense, but you really want to establish memory and innate and adaptive immunity. So what I have on this next slide is a diagram. There's more to it. It's animated, so we're going to go through it together. And what this is my synopsis of how we actually, what we would say, mount an adaptive immune response. In this case, we're going to focus on an antibody response or a humoral response today. That's the one that's covered in that video. And so I'm going to take you through just a general synopsis of what's happening, and then we're going to dig into those subtle aspects as well. And so like each of these big yellow terms, we're going to be actually analyzing those. But there's three things really happening at the same time to initiate an adaptive immune response. So you have to think, first off, let's say you have a cut on your finger. That's usually what I think about when I'm thinking about this process. So you get a cut on your finger, and your innate immune system activates. You bring neutrophils to the site. There's resident macrophages. We talked about these things before last week. And those macrophages and those dendritic cells that get called there, they gobble up all the bad guys that are around. They gobble up self-antigen. They gobble up foreign antigens. And so they're going to grab those and present them on their surface. When they do that, they then head to the nearest lymph node. Okay, later on in this class, we're going to actually talk about how do they know where to go? That's a really good question. It turns out they have special receptors, special homing receptors on their surface that adjust. And so the lymph node is secreting, it's almost like a tasty smell that they can smell. And when the macrophage gobbles up a bad guy, it changes what things it can smell. And all of a sudden, lymph nodes smell delicious and they head towards the lymph node. That's really not that off for how it actually works. So at the same time, B cells are also circulating through, mostly through the blood, but maybe they end up near where that cut is too. But you're going to kind of assume that if it's a pretty severe infection or cut, that bacteria, let's say, has entered the bloodstream, okay? And so some of the antigens on that bacteria are going to be recognized by an antibody on a B cell. So B cells not only secrete antibodies, that's kind of their end goal, but before they do that, they just kind of walk around with the antibodies on their surface and they kind of test them out. And so next week, we're going to talk about how B cells and T cells, but how B cells pick which antibodies to use. So we're going to talk about the antibody structure this week, though. And so antibodies, B cells put antibodies on their surface, and that antibody on that single B cell, okay, is incredibly specific. Okay, it can only grab onto one epitope on one antigen. We're going to talk about what epitopes and antigens are in a minute, but it's only incredibly specific for that one antigen. Okay, and so next week we'll talk about how we get rid of B cells that attack you. But normally we're going to say that we got rid of all the ones that attack you, and they're only the ones that are left that can attack foreign antigen. But there's potentially billions or trillions of foreign antigens. And so that's really what your body does. It makes every possible antibody combination for every possible antigen in the known universe. I'm not exaggerating. Now, is that inefficient? Yes, it is. But we don't have a better way to do it. This is how we evolved to do it, so that's the way we're going to do it, okay? But you just have to take my word for it that there will be a B cell somewhere floating in the blood or the tissue or wherever, and that one B cell out of the millions that are floating around has an antibody that will recognize a surface antigen or one of the epitopes on one of the antigens of this bacteria that's entered our cut. So, last thing, we also have T killer cells and T helper cells that are floating around. Those are CD8 lymphocytes, those are CD4 lymphocytes, respectively. We're going to spend some more time talking about T helper cells a little bit today and T killer cells next week, and how they actually make their surface receptors. But suffice to say for this, we're just going to assume that they're going to end up heading to the lymph node. So if your macrophage gobbles up a bad guy and heads to the lymph node, and your B cell gobbles up a bad guy and heads to a lymph node or recognizes it with its antigen and then gobbles it up, they head to the lymph node, and then the lymph node's freaking out, and so it calls the T cells. And so basically, the lymph node is the super cool place to hang out after you've encountered some kind of antigen. And we're going to think about it as a foreign antigen. Okay. Now, something to start thinking about, though, is what if your body is accidentally treating one of your antigens as foreign? Okay, so one of your self-antigens as a foreign antigen. Well, now you have autoimmunity. And the same process would occur, it's just targeting one of your antigens, which, of course, is a bad thing. But next week, we're going to talk about how we prevent that from happening for the most part, because obviously autoimmune diseases still exist. Anywho, so let's come back to this diagram with me, okay? So we've got our macrophage, gobbled up a bad guy, heads to the nearest lymph node. We've got our B cell, grabs onto a bad guy, sucks it inside of itself, and heads to the nearest lymph node. We've got CD4 T cells, those are the ones we're going to focus on. They're heading to the nearest lymph node because they heard that there's a party happening. And so, let's take a look here. So, in this case, this would be like your macrophage or your dendritic cell, whatever your professional antigen-presenting cell is, okay? And so I'm going to call them APC. And we've got our APC right here. We're going to be ignoring CD8 T cells down here for the most part, right? At least for today. We'll talk about those more next week and maybe the week after that. And so we're going to look here, and there's one of those T helper cells. So all our T helper cells are heading into this lymph node because they heard it's a cool place to hang out, but they don't all recognize the antigen that's being presented by the antigen-presenting cell. Okay, there's only one T helper cell that can recognize that. Of the millions that are in that lymph node, only one has the special T cell receptor that actually binds to MHC class 2, which we're going to talk about, and the antigen. So see that little green thing right there, that little green ball? That's the antigen that's getting gobbled up and presented on this macrophage, let's say. Okay? And so the macrophage is going to talk to that T helper cell. He's going to activate this one specific T helper cell. Remember, he's very specific. There's millions of T helper cells and only one has the ability to recognize that. That sounds really similar to that B cell I was talking about. Interesting. I wonder if the T cell receptor and the B cell receptor, or antibodies, are similar. More on that next week. So we now have a macrophage that has entered the lymph node and is presenting a foreign antigen. We have a T helper cell that has now recognized that foreign antigen using its T cell receptor, or TCR. So now we need to move on and follow that B cell. So that B cell is going to come down here, and our B cell, let's say that it arrived a little early to our party. Let's say these guys aren't even here yet. Our B cell just used its antibodies and grabbed onto a foreign antigen and sucked it inside of itself and presents it on MHC class 2. B cells are also antigen-presenting cells on MHC class 2. And so when the B cell does that, it just hangs out in the lymph node and waits. Which you should ask yourself, why is it waiting? B cells are supposed to make antibodies. There's a bacterial infection going on. Why is that B cell just sitting there? Well, that's a great question. And it comes back to the idea of having self-antigen or generating autoimmunity. So like I just said a minute ago, or let's say you have a B cell that gobbles up an antigen. What if that was a self-antigen? Well, that would be really bad, right? So if that B cell started making antibodies against a self-antigen, now all of a sudden you have an autoimmune disease and you're attacking your own body's cells. Well, that would be really bad. So our B cells, our adaptive immune response to make antibodies through B cells, has a check and balance. So our B cell is going to head to the nearest lymph node and wait. They enter a state that I like to call primed. So the B cell used to be this naive lymphocyte. It didn't know anything. We trained it up. It's ready to recognize a foreign antigen. That's what its antibodies do. But it's going to kind of walk around and it's looking around through the blood, waiting for its antigen. Most of them, they never see their antigen. But this one just happened to. This one recognized that bacterial antigen, and so it heads to the nearest lymph node. It doesn't necessarily know that this is a foreign antigen. It could be a self-antigen. The B cell doesn't care. All it knows is that it's recognized an antigen and sucked it inside of itself. And so what it's going to do is it's going to wait for the proper signal to come from its helper, its friend, its T helper cell friend. And so there's a T helper cell that needs to bind to this B cell that's been primed. It's ready and raring to go. It wants to make those antibodies. It's ready to do it, but it's going to wait. But it needs T cell help to actually generate antibodies and become a plasma cell. Now, I'm being a little dramatic. I don't know why. I just like talking about this stuff. Plasma cells are the cells that actually secrete antibodies. All B cells put antibodies on their surface. We call them B cell receptors. It's dumb. I don't know why we call it that. They're antibodies. But plasma cells are what actually secrete it. So the whole point of all of this, if I back up just a second, is to get a B cell making antibodies against a specific antigen. But you don't want it making it against yourself. We get rid of most of the B cells that will attack you, but it's not perfect. And so we need to make sure that there's some kind of check and balance. And so that's the job of the T helper cell. T helper cell's job is to give B cells the go-ahead. So a T helper cell has to get activated by a dendritic cell or a macrophage that also has that exact same antigen, okay? So here's the connection. Here's my T helper cell. We know that this T helper cell will only bind to that specific antigen, that little green ball right here, that's presented on this macrophage in MHC class 2, okay? So this T cell receptor is incredibly specific. That is like an understatement. It's absolutely, like, there's like only one of these in your whole body out of the millions, okay? Just to be slightly exaggerated. It's really not that exaggerated. And so, to back up just a little bit, we've got our macrophage, and he says, hey man, I'm looking for a B cell. I found this antigen. It might be self, it might be foreign. If it's foreign, that's real bad. If it's self, we don't want to attack that. But if it is foreign, we need to make a B cell response. And so the T helper cell says, oh my goodness, I have a very specific T cell receptor that only grabs onto MHC class 2 and this little green antigen piece right here. And it says, okay, I will do your job, macrophage. So the macrophage says, all right, T helper cell, go look for a B cell. And so he goes, all right, I'm going to go look for a B cell. If I find a B cell that has the exact same MHC class 2 with that exact same antigen in it, I will recognize that with my T cell receptor, and I will activate that B cell. And so he goes looking through the party, blah, 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 blah. Oh, hey, Steve, how are you? Oh my goodness. Is that you, B cell? Do you have an MHC class 2 with this exact same peptide? I should have changed the color to green, sorry. It's the exact same MHC class 2 with the peptide. Oh my goodness, it's time. So the T helper cell that has been activated now activates this B cell, and the B cell freaks out. He says, all right, I need to become a plasma cell, and I'm going to then make tons of antibodies that grab onto this antigen, and we will now flood the battlefield with all this delicious antibody. And now my macrophages and my neutrophils and all my cells can now come in and use these delicious antibodies as a guide to help us attack this foreign antigen. So you can tell I get a little into this. I get kind of dramatic, but it helps me to tell the story. Now, we're going to come back to this chart a lot. As I said, there's actually a whole section that we're missing, so I'm kind of ignoring the T killer cells. We're going to talk about those. T killer cells are responsible for destroying virally infected cells, because virally infected cells, they're like virus factories, but they don't have space. Like antibodies can't get into virally infected cells, so we can't use antibodies. So we just have to blow up the factory, which is pretty cool. And so to do that, we have to use a different process using MHC class 1, which we're not going to talk about today. But this is going to be the same chart that we're going to keep on using. And so that's the gist of what's happening. So now I want to kind of delve into some of the subtleties of these specific processes, okay? The first one being antigen-presenting cells engulfing pathogens. First things first, I mean, we need to talk about pathogens and antigens and epitopes, okay? The gist of it is that a single antigen can have multiple epitopes, and an epitope is defined as a portion of an antigen that can be bound by an antibody. That's the definition of an epitope. And so we say that lots of antigens have multiple epitopes. HIV, for example, the membrane-bound proximal external region of the GP41 on HIV has like 4 or 5 different epitopes just in one small, like, I don't know, I think it's like 100 amino acid chain for that protein in HIV. It has like 5 or 6 spots where different antibodies can grab onto, and each of those would be called an epitope. And some of them actually have some overlap too, which is really cool. And so now that we understand what an antigen is, we need to talk about, we're talking about APCs, or antigen-presenting cells, like macrophages or dendritic cells, gobbling up these antigens, right? And so we're going to jump ahead and you can see, so here's just the basic idea of how a macrophage would gobble up a bad guy, and then we need to chop up the bad guy, chop up all of its antigens, and present those on MHC class 2, okay? So today we're talking about MHC class 2. That's what antigen-presenting cells use to present foreign antigens that they've gobbled up. So professional antigen-presenting cells use MHC class 2. MHC class 2 stands for Major Histocompatibility Complex 2. MHC class 1 we will talk about next week. Similar structure, similar idea, but those are on every single cell. But antigen-presenting cells, or professional antigen-presenting cells, as they're usually called, I'm just going to call them APCs, like macrophages or dendritic cells, use MHC class 2. And the job of MHC class 2 is to present foreign antigens, just like this. And so they get digested, they get placed into the groove of MHC class 2, and then sent to the surface of the macrophage. And then the macrophage is then going to head to the nearest lymph node, or we call it traffic to the nearest lymph node. So we're going to jump ahead now, and we're going to talk about how the B cells actually pull in the antigen, and it's super, super similar. So normally we think about like our macrophage as an antigen-presenting cell. It's going to use phagocytosis to gobble up a bad guy. Well, B cells are actually kind of similar, except instead of using phagocytosis, they will recognize a pathogen using an antibody on their surface that is incredibly specific to that antigen, okay? So it's incredibly specific to an epitope on the antigen on the pathogen, okay? So they're saying pathogen here, really it should be like antigen, and then this would be an epitope that the antibody is binding to. And so we call them B cell receptors. They're just antibodies on the surface, okay? And so they recognize it, and actually typically you need 2 of the surface antibodies to recognize it, and then you have what we call dimerization, when 2 of those antibodies bind and they're physically close enough together on the cell surface that it triggers a response, or it triggers a special pathway that causes the engulfment of the pathogen or whatever those antibodies are bound onto. So once that pathogen is brought inside the cell, it's digested. So it's all a similar process to what happens inside of a macrophage. Okay, you're going to bring it in, you're going to take your pathogen, just like in a macrophage, you're going to chop it up into little pieces, and you present it on MHC class 2, because MHC class 2 is the MHC that grabs onto T helper cells, that grabs onto the actual marker for T helper cells, which is called CD4. So you'll hear T helper cells called CD4 T cells or CD4 positive T cells. Well, CD4 is just a marker on T helper cells that helps grab onto MHC class 2 as opposed to class 1, which we'll talk more about that next week. But so that's where that comes from. And so B cells do the exact same thing. But this all makes sense because if we come back to our original picture, remember, our macrophage uses MHC class 2 with that foreign antigen and presents it to a T helper cell. That's what T cell receptors on the T helper cell are looking for. Okay, they're looking for that MHC class 2 with that foreign antigen. And look, hey, there's that CD4. It only binds to MHC class 2. And then that CD4 positive T helper cell is now going to look for B cells, right? And it's looking for that exact same MHC class 2 with that exact same antigen, and it's going to try and look for it and try and find it. And so that's what we're seeing here. This exact same thing is happening. And so B cells need to use MHC class 2. And so what a B cell is kind of thinking, this isn't how they work, they don't actually have brains, but how they're kind of thinking is, I'm going to grab this antigen that I recognize, okay, so that's happening, okay, but I am a very powerful weapon. And I need to wait until I get the go-ahead from my helper buddy, my T helper cell friend. And my T helper cell, he's kind of a goof. He can only recognize MHC class 2. So I need to take my foreign antigen and put it on MHC class 2, and then go wandering around the lymph node looking for a T helper cell that also recognizes this MHC class 2 using its T cell receptor. If I don't find it, I die. B cells, if they don't receive this signal, they're never going to make antibodies, okay? So that's really, really good, because what that means is either I have gotten rid of the pathogen so fast that there are no T helper cells that are undergoing this process, okay? So maybe my T helper cell never actually got activated by my macrophage because we cleared out the infection, and then this B cell is just kind of pointless and it just dies by, it atrophies, or it would undergo death by neglect is what we typically call it, because it's not receiving a positive growth signal. But if our T helper cell does receive that signal, then he's going to tell that B cell that I like to say it was primed. That's not typically what we would say it, but I like that term. So that B cell was ready and primed and ready and raring to go, and now it received that signal, and now it can convert into a plasma cell. So the next thing I want to talk about is just, we're going to dig a little bit deeper into how these cells are actually touching each other and talking to each other. And that kind of needs a little bit more context for the actual structure of a lymph node. Now, lymph nodes are considered secondary lymphoid organs. And so we've all heard of lymph nodes, like usually we think about your lymph nodes that are sort of like under your chin there, because those are typically the easiest to find if they're swollen because you have some kind of infection. Let's say you got an ear infection or like a throat infection or something, and your lymph nodes are swollen. There are lymph nodes all over your body. And we're going to talk a little bit more about secondary lymphoid organs like that, as well as primary lymphoid organs next week. But this week, I really want to hammer home this idea that you're going to have these three things come together to come hang out in the coolest place to hang out ever, the lymph node. And so let's actually look at the structure of a lymph node. And so here would be a typical lymph node. We're going to say it's secondary lymphoid organ because they're not all lymph nodes. They just have different names, but they all have similar structures. So we're just going to kind of assume they all look like this. And so down here, you can see here we've got our antigen presenting cell. So this is kind of like from the other diagram. And so we've got our antigen presenting cell, and it's going to activate a T helper cell here. Okay, so he becomes activated, and then he goes looking for a B cell that is also presenting that exact same antigen on that exact same MHC class 2. And so inside the lymph node, we've got our germinal center here, and here's our B cell. Okay, and so this is our B cell that came from the other diagram as well. Okay, so this is our B cell from here. So he was hanging out in like the blood or the tissues or whatever and found that foreign antigen and recognized it, and then headed to the nearest lymph node. So that's there, he recognized it, heads it into the nearest lymph node, finds the antigen, and is presenting that antigen on MHC class 2 to our T helper cell. And so now it has become activated. There are a couple things that happen. Even if you only had one B cell that bound and actually had this happen, the odds are stacked against an immune response actually happening. There's millions and millions of B cells, there's millions and millions of T helper cells, and only a couple, if there might be one that can actually recognize and trigger this process. But let's assume that it happens. Well, we need to get a bunch more of those. And so your B cells immediately start growing and proliferating. Your T helper cells do too. And so why would they do that? Well, because we want to really amplify this entire process. And so we're going to make tons of these cells because we're like, oh my goodness, this B cell with this specific antigen is awesome. We know that it attacks that specific antigen. And so I want to make a ton of them. So we have B cell proliferation is the term that we would use for cell growth. We're also going to have a ton of T helper cells grow. And these B cells are then going to go on to, this specific diagram is a little complicated, I know, but early on they're going to start making antibodies, and then you have some memory cells, okay? So those are the two main routes that we're going to go. We're going to have plasma cells, which are just B cells that are secreting antibody, they're doing their job, okay? And then some of them become memory cells so that the next time they encounter an antigen that they recognize, they don't have to do this process. So they don't need T cell help to actually activate and start mounting adaptive immune response. And so that's why vaccines work, or that's why you don't get the exact same cold again, or the exact same flu strain again, because you have these memory B cells hanging out and they can instantly become activated without T cell help. There's something really, really interesting, and it's the last thing I want to talk about today. And what that is what happens to a B cell after it becomes activated and it starts proliferating. It does this really interesting process called clonal expansion and affinity selection. Okay, so you can see these here. So we have clonal expansion and affinity selection. And I want to talk about these processes. They're really interesting. They sound super cool. And I remember when I was first learning immunology, I was like, oh man, clonal expansion. Clones? Like Attack of the Clones? Affinity selection? I don't even know what that means. Well, I'm going to explain it to you in plain English. So we're going to ignore light zone and dark zone and follicles and germinal centers and all this crazy stuff. Here's what I want you to know. Only the best survive. So when we take our B cell and we have activated it using our T helper cell that was activated by a macrophage, and we are going to take that B cell, and let's say there's only one, and it starts making tons of itself, okay? It's proliferating. Those are all identical clones to the original, okay? They're undergoing tons of mitosis. They're just replicating like crazy, and they're all identical clones. Now, that would be great. Like, they're going to make antibodies that bind to this foreign antigen. That's awesome. But our bodies are really, really cool and really interesting. And what they can do is they can say, all right, we know that this B cell is awesome. And so what we're going to do is we're going to take all of those clones of B cells, okay, there's hundreds and now there's millions, and we're going to take all those clones and we're going to send them through a couple different processes. We're not going to go too deep into them today. One is called somatic hypermutation. And another one is called class switching. Basically what we're doing is we're going to take those antibodies, because that's the business end of the B cell, right? Is those antibodies. Those are the weapons that they're going to shoot at these bad guys and stick to them and kill them. And so these antibodies, the business end of those antibodies, the little grabby ends of the antibodies, we can change them using a process called somatic hypermutation. It's where we go in using a special enzyme and we mutate the genes, which should terrify you. Why would you want to purposely mutate a gene? Well, here's like the one case where you would want to do that. And so we modify the little ends of the antibodies just a little bit to make them just the slightest bit different. And then we let it keep on growing, and it makes a pool of B cell clones. They're not all identical, but they are still clones. And then we sort of challenge them against this exact same antigen that we've been trying to come up against and fight. And then we pick the winner, okay? So only the best one gets to go on and survive. So let's talk a little bit about how that works. Basically, so here's that same B cell, okay? There's that initial one, okay? That's this B cell that we're looking at right here. It cloned itself, it grew, it proliferated. We're going to ignore some of these names. The names aren't important, but we're going to take them and we're going to modify those antibodies. We want to see, hey, if I change it, if I tweak it this little bit, does that make it bind better to this foreign antigen? Because that's the name of the game. I want my antigen to be bound so tight that my antibody can never let go, and that will stick to it forever and allow all my macrophages and neutrophils and all those awesome cells to find them super easy and to kill them. And so that's what we see here. So in this case, we mutated our antibody and it didn't work better. It actually worked worse. And so we get rid of the cell. And that's happened again down here. But these two cells, okay, these two B cells, they modified the ends of their antibodies using somatic hypermutation, and it actually worked better. Well, how do they know that? Well, there's a special cell called a follicular dendritic cell, and there are T follicular helper cells. Their names come from the fact that they're in the follicle. That's what these are called. I know. These terms are awful. So they live inside this special place where this is happening inside the lymph node. That's where their name comes from, okay? They're not really that magical or special. Like, that's just where they live. They live in the follicle, so they're called follicular dendritic cells. And so their job is to actually test these B cells. And so they'll take this foreign antigen and they'll like poke it at the B cell and see if the antibody grabs on it. If it binds really tight, they receive a special signal that says, hey, you're even better than the last version of yourself, you crazy clone you. You should become a plasma cell and start secreting antibodies. And this process repeats itself over and over and over and over and over. Every time there's that foreign antigen present, you're doing this process. And so let's say you got a bacterial infection, you clear it up, and then 2 months later you get a cut on your finger again, and your B cells they have memory B cells, okay, so they're ready and raring to go. They don't need that T helper cell help. They become activated immediately, but guess what? They still go through this process and become even better at binding that foreign antigen. Why is that great? Well, guess what vaccines are? Hey, you need a booster vaccine? Yes, because every time you get a booster, it's going to make that antigen bound better and better and better, okay? So your B cells are better every single time you get a vaccine. So I have another diagram here that just kind of illustrates this idea. So we've got our clones here. They came from that original activated B cell, and you would make some cells that they're always secreting antibodies, and then you have some that become memory cells, and so you've got that memory cell just kind of hanging out. Let's say we get rid of that foreign antigen, but then it shows up again, and then we start our proliferation again. We don't need T helper cell help, but we can proliferate and make all these, and these cells also undergo that selection process. I talked earlier about somatic hypermutation. That's how we get our antibodies looking different, okay? And we may talk about that later on in this class. But something else I talked about was class switching. And we need to talk about what class switching actually is. Now, when you first start out, when you're a B cell, you express your B cell receptor, which is just an antibody, and you express something called IgD. Ig stands for immunoglobulin. That's just the name that we give antibodies. So antibodies, a body, they knew it was something, so they called it a body, and it was anti-something. They knew it was something that bound to something, so it's anti-something. So we also knew that it was a soluble protein, and so we called them immuno because we knew it was immune-related, and it was globulin because we knew it was a protein that had a globular structure. And so that's where the Ig comes from. And you'll see this for medications, they'll be called like immunoglobulin therapy, and that's where that comes from. Okay, so your B cell, the first antibody that it puts on its surface is called IgD. It's very early on. The actual grabby portion, okay, that's not the technical term, but the portion of the antibody that grabs the bad guys is not super tightly binding, okay? But hey, it works all right. And so that's not the kind that you want to secrete, though. The kind of antibodies that you want to really get out there, you want your IgMs getting out there. These are even better at grabbing stuff. And IgG is like the king of grabbing onto foreign antigens, okay? And so we start out with IgD. We move to IgM, okay? And this is all through this clonal selection process. So over time, as this process is happening, as we're going through somatic hypermutation and we're just selecting our B cells that bind onto antigens, not only are we modifying the business end, but we're also sort of changing which antibodies we're picking because they can have different functions. Okay, so early on we want our IgM. So you can see this is a pentameric antibody structure. It has all these extra binding sites, and so you can really grab a lot of antigen that way. but you really want to turn into IgG, and that's what we see later on during an immune response. IgG is sort of the antibody that you want to head towards. IgG is the one that has a really, really specific binding site here on these grabby portions of your antibody. And so that's the antibody that you're heading towards. Now, if you happen to be fighting a parasite, well, our bodies know that parasites are really weird and kind of scary. And so instead of using IgG, they can switch to IgE. So this is the kind of antibody that we see. You can see it's similar structure to IgG. So they're both very powerful and really specific grabby parts here. But IgE looks a little different because we know that parasites are really scary. IgA is a little bit different. You can see it's basically like two IgGs stuck onto this crazy looking central portion, this secretion protein here. And IgA is basically what the type of antibody we put on mucosal surfaces. And so it's like taking two IgGs and getting two for the price of one, and then you put it on a mucosal surface. But really, the two antibody classes that we're mostly concerned about is IgM and IgG. And they all serve different functions. So on this next slide, I have our different classes and the different functions of the classes and the different sizes of the classes and how much of the total serum of the classes. And you can see here, 80% of the antibody in your blood is IgG, because usually if you're encountering foreign antigens all the time, you're encountering the same ones over and over again, and so your body gets really good at making antibodies that grab onto bad guys, and so that's IgG for the most part. Fun fact, IgG can also cross the placental barrier. It's the only antibody that can do that, and so that's a way that the mother can give the baby antibodies to fight off infection. Or IgG is also transferred through breast milk, and so that's another way that mom can give baby antibodies to help fight infection, which is really, really cool. We'll talk a little bit later in this class about complement. You can see here, so Fc binds to, that's referring to is antibodies have the Fc portion, that's this end of the antibody, so not the end that grabs stuff, but this end is called the Fc portion. And so there are Fc receptors on all of your immune cells. So like your macrophages and your dendritic cells and your neutrophils and your eosinophils, all those cells, they have Fc receptors, and those are very specific to specific antibody Fc regions. And so when they talk about B cells, and so, like in the video where they talked about the antibodies are sticking to the bacteria, and then macrophages like to find those and use special receptors to grab onto those, they make the bacteria look delicious. That's the Fc portion. So it's the other end. So this end is grabbing the bad guy or the bacteria, and this end is the end that binds to specific receptors on immune cells. And so that's how we're getting this synergy now. Okay, so we had our innate immune system. It was helping to fight the bacteria, but I don't know, it's not always perfect. It needs help. And so it sends a macrophage to the nearest lymph node. It says, hey, T helper cell, you got to find a B cell to help me. And the T helper cell is looking around. He finds a B cell to help him. That B cell clones itself. It makes tons of itself. It makes tons of antibodies, and those get secreted. And those antibodies bind onto the bad guy. And those antibodies themselves come back and bind to the macrophages through a special receptor and help them eat the bad guys. And so that's how antibodies are actually helping. So we should talk just a little bit. I want to give you a little bit more detail about antibodies and antibody structure. Their names are tossed around a lot. And so you'll hear a lot of these terms tossed around: light chain versus heavy chain, constant region versus variable region. Those are really the two, the four terms that get tossed around. And so you can see, I love this picture right here because it really distinguishes what's going on, okay? So antibodies have like two halves, right? And each of those halves has a really long chunk and a shorter chunk, okay? So the short chunk is called the light chain, and the longer chunk is called the heavy chain. Okay. And on the end here, part of the heavy chain and part of the light chain is the variable region. And this is what I call the grabby part, or the business end of the antibody. This is the actual portion of that amino acid chain that grabs onto foreign antigen. So the rest of this is the constant region, or also called the Fc region, which I just talked about. And so that's the region that will grab onto or act as a bridge to receptors on macrophages or granulocytes or whatever. So the last thing I wanted to talk about today was actually the different functions of antibodies. So I was talking about how antibodies can grab onto a bad guy and make them look delicious, quote unquote. That process is called opsonization. And you can see that here. So opsonization is where it coats a bad guy. So in this case, it's a virus. And then it's bridging the gap between the foreign antigens and the actual macrophage, let's say, or a neutrophil, and using Fc receptors, okay? And so that's called opsonization. Now, antibodies themselves can directly neutralize bacteria, viruses, or whatever, usually by forming aggregations. And so, like you can imagine, antibodies have two grabby pieces, and so one antibody could grab onto one virus, and then its other grabby arm could grab onto another virus, and now those two viruses are stuck together. And if you have millions and trillions and quadrillions of antibodies floating around, you can really get these giant globs of gross neutralized microbes, and they just can't move, which makes it really easy for them to be phagocytosed as well. Another thing that they can do is called antibody-dependent cellular cytotoxicity. And so that's basically a fancy way of saying that antibodies, just like opsonization makes them look delicious, well, antibodies can also make foreign cells or cancer cells look delicious to natural killer cells, whose job is to go and blow cells up. Which we talked about with the innate immune system. Antibodies can also help complement. So a complement is a series of proteins that are used to basically complement the rest of the immune system. And we'll talk about them at a later time. But basically, antibodies can trigger complement. And complement itself can do a variety of the things that antibodies can do, like opsonization, or they can directly neutralize or lyse bacteria. They also tend to cause inflammation. They're very powerful, but they complement the rest of the immune system, hence their name, but they also serve a lot of the similar functions as antibodies. So last but not least, I wanted to leave you with just more of a synopsis of class switching, because I sort of glossed over that a little bit. So we can take our B cell, it starts out IgD, it becomes IgM as we're going through that somatic hypermutation, and our body gets better and better at figuring out what kind of pathogen is actually attacking me. Is it a bacteria? What kind of bacteria is it? Do I need to make IgG1? Do I need to make IgG2? Yes, there are subclasses of antibodies, as if this wasn't confusing enough. Also, they have different names between mice and humans, so that's super annoying. But something I wanted to just kind of reiterate is that over time you switch classes. You go from IgD to IgM to IgG, and then sometimes onto like IgE. And so what happens over the course of an immune response, and so when you first get that infection, after a while, your innate immune system, it goes through this whole process that we've been talking about today, and it says, hey, you need to switch from IgM, that's the first one that you're going to make, and you need to start making IgG antibodies. And so that's that class switching that occurs. And this would be the first immune response that you see, or the primary immune response. So the first time you get like a vaccine, this is what your antibody response looks like. And then as the antigen in the vaccine starts to clear out, then the antibody response starts to go back down. Or if you get the flu and you don't have a vaccine against it, the first time you encounter that flu, this is what happens. And then you're eventually clearing the flu virus, and then the antibodies can go away because you cleared the virus. But class switching occurred. And that secondary immune response is much, much bigger. And we'll talk about that later. So what we're going to be covering next week is going to delve a little bit deeper into B cells. So if this is tickling your fancy a little bit, well, stay tuned. We're going to talk a little bit more about B cell genetics and T cell genetics, and how B cells and T cells are actually trained, because I glossed over the fact that these B cells and T cells are very specific to the antigen that we're trying to target, right? And so they're very specific to those antigens, that's fine, but it turns out that when we actually make our B cells and make our T cells, we make every possible antibody, every possible T cell receptor that can bind to every possible antigen, including self-antigens, in the entire universe.